Hello, everybody. I want to talk to you today about our studies from origin of life to eco-friendly next generation medicine and fascinating viral vaccination, which is now very, very much in fashion because of the new vaccination for the COVID. So let me just start my screen, share a screen, and hopefully within a minute we start. So we share this one. And I hope that you can see something. So as I told you, from origin of life to eco-friendly next generation medicine and fascinating viral vaccination. And I'm very proud to give this lecture because origin of life for me is the story of the egg and the chicken, which was also the story of the late Professor Schrödinger. So the unified process of DNA to protein goes through transcription to messenger RNA and translation by ribosomes to proteins. And this is a universal process. Of course, now messenger RNA is in favor and in the news. As I say, because at least two vaccinations are built of it, and I'll come back to it. So first about ribosomes. There is a huge number of them in every living cell. And mammalian cell may contain millions of ribosomes. Let's say in the liver can be 6 million of ribosomes. Even in bacteria cells, there are up to 100,000 ribosomes. They are performing two major functions. Decoding, they read the code and they make the peptide bond. It means they make the protein. And they do this in the same way, no matter where the ribosome comes from. Bacteria, elephant, flower, whatever. They act continuously. They can form 40 peptide bonds in 1 second. I needed a few hours in the lab when I tried to do it myself. And they hardly make mistakes. Mistake rate is between half a million to 1 to 1 million. So it took us 20 years, but at the end of the 20 years, we got the structures and immediately we understood the function. So here is a little video, Here is a little video made by two students of the Art Academy in Jerusalem that shows how bacterial ribosome works. But now it doesn't start. So what you are about to see here is how the messenger comes into the ribosome. Here, the cell is green and the messenger is this line. And it comes to the small ribosomal subunits. All ribosomes are made of two unequal subunits, small and large. The small hosts the messenger. And this is possible to be accurate by factors, initiation factors. Even bacteria have 3 of them. And this is factor 3, initiation factor 3. Once messenger is in place, the first tRNA carrying the amino acid can come with initiation factor 2. Now the factors have left, everything is in place. Here is this little thing is the amino acid, and the large subunit can come and associate itself to the small one. So now the ribosome is ready to work. More and more amino acids are being brought by tRNAs with elongation factors. The factors are very important. And the ribosome helps in out. Decoding happens in the small subunit. And you can see here. Here we took away the large subunit. We left only the tunnel through which the protein comes out. So the protein is made in the large subunit and goes into the tunnel. So now you can see the whole ribosome again. Protein starts to be large and comes out. Folds alone or with chaperone, we won't go into it now. And the process continues and continues until there is a stop codon. And then instead of tRNA come factor, release factor, recycling factor, this gold thing here. It comes in, it separates the two subunits. The protein can come out, it's finished. And the whole system is waiting for the next job. That's all. So you saw earlier the ribosome is something fuzzy, but actually we know every single atom. This was back in 2000. Now there are many more structures of ribosomes, even larger. They're all made, doesn't matter, larger of human and so on. They're all made of RNA, ribosomal RNA, and ribosomal proteins. The RNA is in gray, the proteins in many colors. And to remind you, the small one makes the decoding, perform decoding, and the other, the large, make the peptide bond. So if we look at it from another view, here is the small and the large. They are connected by lots of contacts between them, but also by the tRNA that is doing the decoding by codon-anticodon interaction here. And the lower one makes the peptide bond in the PTC, peptidyl transferase center. And this is universal. So actually what you saw here and in the movie, small subunit up top, large subunit below, messenger binds to the small one, and amino acids are being brought by tRNAs to the ribosome. There are 3 sites. First site A, where the amino isolated tRNA sits, peptide bond is made in the second, and the peptide is coming out. That's the P site, and E, the exiting. And this all is universal. So again, let's have a look. Small one in pink, large one in gray, in green and blue, and the active sites are here. Each of them has 3 sites for tRNA. So when we looked at the structure, we saw that for all substrates, all proteins, it's always that the ribosome provides a stereochemically frame in a semi-symmetrical position. You can see here, semi-symmetrical, and better you can see when you look at the tRNAs, the blue in the A site and the green in the P site, and I will keep it like this. And the peptide bond is being made here. The newly born protein will go out through the tunnel, as you saw in the movie. So even in the beginning of our studies, within 1 or 2 years after we got the structure, this took 20 years, but I won't talk about it now. Even in the beginning, we saw that all the substrates are sitting at the active site in semi-symmetrical. Doesn't matter if they're very, very large, or there is already a little bit of newly born protein. They are making this pocket type semi-symmetrical arrangement. And please pay attention, it's not only our structures, it's also from other places, for instance, here from Yale. So the semi-symmetrical pocket is actually made of 2 sides, and I put them here now instead of the side, one on top of the other. The A is in the A side of it is in blue and the P in green with the substrate, and you can see how symmetrical, semi-symmetrical there are pieces here and there that are not the same, how nicely it hosts the substrate. You can see from top that we put together all the backbone of all the pockets in archaea, in bacteria, in eukaryotes, even in mitochondria, they coincide. Exactly. the coincide. If we look at the backbone, especially coincide, and this point is the center. So we propose that the high conservation of the structures of the symmetrical region, which is completely conserved, indicates its existence is beyond evolution. So actually what we propose, that the probiotic bonding entity that is termed by us the protoribosome is still functioning in contemporary ribosomes in our bodies too. We assume that the ribosome originated within an environment which could contain elements of life, and that the prebiotic chemistry could generate and elongate RNA chains. So one of our, and this was actually shown in papers, not ours, but in several, we don't have time now. One of our hypotheses suggests that the catalytic protoribosome is a product of dimerization. This requires existence of self-replicating, self-folding, and self-dimerizing RNA molecules. And we tried to make it in the lab. It took us a long time, more than 5 years, just to find out how to make a protoribosome, but we were able to make it. Not only this, we made some protoribosomes, and we showed that some of them, not all, but some of them that have the sequence that was expected to be fruitful could make new bonds, new peptide bonds. It means we showed that this could be real. So to summarize, our hypothesis is based on the existence of self-replicating RNA molecules with catalytic capabilities, and on the assumption, and this is important, that the genetic code followed its product. So now we know what is the chicken and what is the egg. Do we really know? But we think that we understand better how things started. So based on the suggested existence of RNA domain world, and on the experimental result, we propose that the protoribosome is the entity around which life has evolved. So now a few words about ribosomes and their antibiotics. So owing to the vital role played by ribosomes, almost half of the clinically useful antibiotics target protein biosynthesis, mostly by paralyzing the ribosome. Natural antibiotics are the weapons that microorganisms from one type are using to fight against the others. So I never expected, during the 20 years that we worked on solving the structure, I didn't expect that we will see the antibiotics. They're so small, the structure is so complicated. But we do. Here you can see Ada Yonath that was found in Poland that sits exactly in the messenger RNA position, path, doesn't let it go. You see, so small, so clever. Second one is even smaller and even more useful. It's called tetracycline, and it occupies the position of the second tRNA. So one tRNA can bind or even a chain that was earlier there, but now the A site is blocked. That's it. So you notice that both of them are functioning on the active site. The third one, the erythromycin, which was actually the first from the ribosomal antibiotics, right after penicillin, binds just below the peptide bond formation place in the entrance to the tunnel and blocks the tunnel. That's it. Protein cannot go out or can go out with difficulties. And the last one that you will see now is the cleverest chemically. It goes into the active site like that. Just blocks the next amino is coming in. So essentially what you saw now are four antibiotics, but there are many more, and they are shown here. And all of them bind either to active sites or to the paths into the active sites, like here and here. So this is correct for all antibiotics made naturally. But antibiotics are wonderful, but resistance to antibiotics is one of the most severe problems in modern medicine. And although it's so severe, only few antibiotics are in development by the companies. The companies are not producing, not not investigating how to make new antibiotics, almost not. Left, the speed of the appearance of resistance in three different antibiotics. So sorry, three different cells, three different bacteria. So you see here from 1990 to 2002, just 12 years, how fast the resistance grows. And on the right side, you can see how little. This is from 1984 to 2014, 30 years. And that's all the new antibiotics from all the companies in the world. And the last one, as you can see here, is about 6 years ago. So we asked ourselves, can we combat resistance to antibiotics in full? And in my opinion, it's unlikely because bacteria want to live and they are cleverer than us. Yet, we have the concept how fighting bacteria's wisdom, identifying pathogen-specific essential structural motifs that do not belong to the active sites. We will not go back to what bacteria do, we look for new ones. So our modest contribution was to determine the structure of ribosomes from resistant pathogens. And we did it for 4 of them and compared them to harmless bacteria. We have many lessons. I won't show you all. I'll just tell you about some. All bacterial ribosomes are similar, but not identical. The lesson number 4 is the one that was beyond my expectation. It hints at possibilities for new antibiotic binding sites. So what you see here is superposition of a non-harmful bacteria in gray and additions of the resistance pathogen in cyan. There are more, there are in and out, but I drew here only those that are relevant to what we saw here. So if we look at one of them in more detail, you see that the neighborhoods are the same for the pathogens and the harmless. But this piece is additional and it goes outside and it is not bound to anything. So it may be a potential binding site. So actually, if we look at all of them, all these can be potential binding sites. So essentially, we identified in a model system 25 new potential binding sites, and we block them one by one chemically, and 16 of them inhibit protein biosynthesis. And since they are not involved in the primary ribosomal activity, neither in decoding or neither peptide bond formation, they can maybe be better for new antibiotics. So I just want to show you that the structure of TB, it means I will remember the name in a minute, versus another pathogen, Staph aureus, and you can see that the tuberculosis is the first name. The tuberculosis has here, and compared to another resistant bacteria, it has here and here several, and here several new pieces. Like those out of those we could find, for instance, this one and this one as a basis for new sites. So for now, these sites can be exploited for the design of advanced degradable antibiotics and we can make them environmental friendly. So let me just say a few words about selectivity and similarity. You know about the microbiome, harmless bacteria that are really good for us. They can be destroyed by the currently used antibiotics, but we can design the new ones that will not hit the microbiome and also will not compete with the eukaryotic, the patient ribosomes, because as you can see here, most of the changes between bacteria and human are either in the red or in the pink or in the green, but on the surface. It means if we talk about surface, we don't have to worry about hitting the patient. So the lesson number 6 is the ecological conditions. Most of the ribosomes, the ribosomal antibiotics that are used today have within them pieces and cores that cannot be degradable and cannot be digested. So they are not digested, not biodegradable. Therefore, they can sneak into the irrigation system because the filtration doesn't always catch them and come back to us through the grass or whatever grow on it. So with us, we can make the new identified potential site. We can make them made of nucleic acid, oligopeptides, whatever we think is right. So they are degradable and they can be optimized in terms of chemical properties, antibiotics action, and degradability. So the last thing to talk about is the intruders or the invaders. These are the viruses. You all know about coronavirus. I don't have to show you a lot, but I want to show you that it's a genetic information is in here, and it's in messenger RNA. So they can skip the step from DNA to messenger. So when a virus hits a cell, the spikes are getting into the membrane, and then it goes through the membrane, and then the messenger is being translated by the ribosome. So actually, although the size, the composition, the complexity, the function encodes by the genome, of all viruses are remarkably diverse, all really rely entirely on protein-forming machines, ribosomes, in the host cells. They go into the host cells, take over the ribosomes, and let's see what they take. So, oops, sorry. So the question that I wanted to ask, do the viruses use the host cells machinery, initiation factors, and so on, what I showed in the beginning? Are they using it to make their own proteins? Or they just jump into the already functioning line, just into the queue? How do they do that? So both things to me are not clear. So first it was shown that in all CoV, SARS, all these viruses, there is a program for frame shifting. It means they can disturb the cell to make its own. Actually, recently it was found, really not long ago, it was found that there is a protein that is called NST1 that stops that blocks the gate for the messenger, the gatekeeper. But if it blocks also for the cell messenger. It means nothing will grow or hardly anything will grow. This is an open question. Actually, it's known that it stops by making a specific messenger RNA turn. I don't really want to go into details. That's what we're looking here, and this is when it stands. Okay, so the messenger cannot continue, but how can it get in? This is still an open question. The question I asked you earlier. Do the viruses use host cells machinery, all the factors, or they just jump into the already functioning line? And with this question, I would like to conclude my lecture. I want to thank the president of the Weizmann that let me work, Michael Sella, a biologist, that let me work after the advisory committee said that it's not clear that Ada will solve the structure of the ribosome, but a project like this should be terminated. He was biologist, and they say that he listened to them because he was biologist, but after his term was over, he was replaced by Chaim Harari, a physicist, and he didn't kick me out either. The whole study was in collaboration with Max Planck Institute in Berlin and with the Max Planck Society. Professor Wittmann, that really wanted to see the ribosome structure, died 10 years before the structure came, but he was very important in setting up a group for us in the Daisy in Hamburg. And when he died, Franceschi and then Fucini replaced him. So I had two groups, one in Hamburg and one in Israel, working in parallel. The group in Hamburg has stopped 17 years ago. And in Israel, there is still a group, and those that have these little green buttons are still with us. So you can see here who did and so on, and I won't go into detail, just let you know that Anat Bashan is the senior scientist. She is doing everything. I'm just here as a picture. She's running the group and the projects and so. And I want to that you look at Tamar. Tamar came 18 years ago for 10 weeks. She's still around, and she had a birthday the day I took the picture. So she baked a cake. Do you see the cake? I'll show you the cake in more detail. This is her cake. And it shows that in my group, ribosomes are considered sweet. I also want to thank my family. You can see here my daughter, who is an MD, and her daughter, who is now studying medicine. But when she was really young, 13 years old, she gave a speech about me when I got the prize in Paris. And she said that I was always with her. She gave me a prize. That's not her prize. That's the medal, the Nobel medal. Her prize is at least as important for me. The grammar of the year is Ada Yonath. And when I asked her which year, and she said, which year? You have to reprove yourself every year. Otherwise, I take it off the wall. It's still on the wall for the last 14 years now. Ribosomes became popular. This is a carnival. He is me and she. You can see here she's dancing. Proteins come out of her. Even young kids became ribosomes. And Kishka, who is one of our most beloved artists and writers, made me head full of ribosomes. You can see here the large and the small ribosomal subunit, fully symmetrical, like the active site. And he thinks that antibiotics should be here, 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 and there. Thank you very much. So, next time I hope to see you in life.